Recording. So I think I'm now sharing my screen and recording the lecture. Okay. Okay, so today we'll go through developmental social neuroscience. Oops. Yeah, that was the video I was just searching for here. So we've kind of got three parts. First of all, I'll talk about the nature and nurture of social cognition. So here looking at how we can pull apart genetics and environment and what kind of mechanisms are at play here. And then we'll kind of go through linearly from infancy through to adolescence, how the social brain develops. So again, covering a lot of the issues we've already covered, but thinking about it in terms of a developmental twist. So first of all, if we think of the nature and nurture of this, so how do we build a brain, first of all? So again, most ideas about the brain is that you've kind of got a blueprint that guides it. Actually, that blueprint is more of a kind of a guidebook or a recipe. So the number of synapses in the brain far exceeds, like by millions, the actual size of the genome. So there's no way your genome has enough information to kind of dictate a very fine blueprint. But obviously everybody's brain looks broadly similar. Your cerebellum's at the back, your frontal lobe's at the front, and so on. So clearly there are broad patternings that are very consistent across people as well. So we have to kind of bear both in mind. But this is the kind of the trajectory of physical brain development. So again, you only start to see the folds kind of appearing in the kind of the months before birth. But you've got at some point rapid acceleration of the number of neurons. So at some points you've got 250,000 neurons being formed per minute at certain early forms of life. And basically what happens is that as these neurons are formed, they kind of form from particular place in the brain, and then they migrate outwards. Sometimes they just migrate out just by the inward pressure kind of pushing them out, and sometimes they're almost like protein ladders that kind of pull them out into certain positions within there from these zones. What you find is that the newborn brain is a lot smaller than the adult brain. Actually, the reason for this is probably more to do with the mother than it is to do with the baby in terms of being able to give birth to big-brained infants. You are actually born with your pretty much your full set of neurons. So what's happening when you're increasing the size of the brain is actually probably not to do with your neurons. It might be, it's probably going to be your glia and the other cells that support them, including adding myelin and so on, which will increase brain volume. So as I mentioned, to some extent, the time you're born is the peak of your brain, both in terms of the number of neurons, but also in terms of the number of synapses. So here on the x-axis is your age from conception, and then you've got your number of synapses per whatever volume. Again, this is measured with MRI. This would be kind of pathological, looking at brains being dissected at different ages. And each region of the brain shows this characteristic peak and then fall, that you have more synapses early on and later afterwards, with sensory regions peaking earlier and the last region to peak being your prefrontal cortex, your lateral frontal lobes, which you can see kind of reaches maturation around adolescence here. So we can't assume that having more synapses is necessarily a measure of efficiency. What might happen during development is that tuning in the synapses you need is the efficiency saving. You're kind of almost over efficient or perhaps noisy, if you will, when you're earlier on. What the actual behavioural correlates of this are isn't clear. I mean, clearly infants aren't more skilled than us, but nevertheless there are some examples where they are. So discriminating phonemes is the classic example where infants can tell phonemes apart that we as adults can't. So if you're an East Asian speaker, for example, Chinese and Japanese infants can discriminate R and L, but they can't when they get to 3 or 4. And that's probably related to that mechanism, although nobody's actually joined those dots up exactly at the level of synapses. As I say, the genes are clearly important for building the brain, but they are somewhat imprecise. We cannot specify the exact structure or shape of the brain. But you can look at heritability estimates. So in the same way as you can look at heritability estimates of schizophrenia and so on with twin studies, you can look at it in terms of the physical structure of the brain. So the total brain volume itself is highly heritable, and this is probably things like the number of cell divisions that you go through in constructing the brain, for example. Whereas things like the cortical surface area is far less heritable, as is the grey matter and the white matter density in particular regions. These are still quite high, actually. What creates the surface area or the shape of the brain is probably not specified in the genes. It's almost as if you've got a kind of a sheet, and what determines the sheet is partly the number of cells in it, but partly the actual tension of the axons. So one idea is that the shape of the brain is not coded genetically. You've got axons that are coded genetically, and it's like having elastic bands. So the shape of the brain is kind of an emergent property of how many neurons you've got, how much packing you've got in the head, and these kind of tensions of the axons. But again, you get these kind of nice pictures where you look at identical twins and fraternal twins. So the measure here is the correlation. So here you've got two twins, and you're looking at the correlation between twin 1 and twin 2. And here you're looking at the correlation between twin 1 and twin 2, where they are not genetically identical. Heritability actually is the difference between these ones and these ones here, okay? We can look at that. And this is the grey matter density that's been looked at here with that. So you can see that there are these general trends. We can look at this at the level of social behaviour. So this is kind of just one of those gift reviews where they've pulled everything together kind of from the last 10 lectures, and they've looked at it with MZ, genetically identical twins in blue, and DZ, so non-identical twins in red. So here, the thing that you're looking for to get a sense of heritability is the difference between the blue and the red. But have a look at some of the things on the bottom. You've got pro-social behavior. You've got bizarre things like infidelity. You've got empathic concern. So again, empathic concern, the values tend to be low. That means that the twins in general don't correlate so much with each other, but actually the genetic difference is quite large between the blue and the red. So the overall size of the correlations between the twins are low, but the difference between MZ and DZ is large. So you've got these kind of relative and absolute effects here. You've got risk, parental warmth, leadership, the trust game, so very specific measures in these neuroeconomic things. Obviously, in general, the blue bars are greater than the red. There is always a genetic influence, but we have to think exactly what that is and try and interpret it. So there is not going to be a gene for empathic concern. There isn't certainly not going to be a gene for infidelity in females, okay? So whatever these genes are, they are going to be much broader phenotypes than are kind of implied here. And that is kind of the real puzzle. This is the same data, but in effect what you can do is you can break it down into two different environmental influences. One is shared environment, so that's what goes on kind of within the family. An unshared environment might be, for example, that twins have different peer groups, fall ill at different times, these sorts of things. So blue are the genetic effects, which are basically the difference between the red and the blue bars. That's how you calculate these. You subtract them. I think you divide by 2 or multiply by 2. But anyway, it's basically related to that. And then in green and red, you can see that most difference between twins is actually not related to the family. And again, there's whole debates about this. So the red is the kind of the family influence there. But these, of course, tend to be measured almost certainly in adults. So maybe the family is important earlier or whatever. So heritability just gives you a sense of that there is a genetic effect involved, but it says absolutely nothing about individual genes and what they're doing. And almost certainly there are multiple genetic effects, particularly when we're dealing with very complex phenotypes, like something like, I don't know, infidelity or risk-taking or whatever it is. So how can we kind of link behavior down to the level of individual genes? Well, how do you get your genes? Well, this isn't a coronavirus test. This is a genetic swab test. So basically, you've got a brush that you scrub away. You're not getting the saliva here. You're getting cheek cells that break away into the saliva that create that. So why is a cheek cell going to tell you anything about the brain? Well, actually, it's the same genetic code in your cheeks as are in your brain, as are in your finger and in any other part of your body. That's the way that genes work. When we look at epigenetics, that's different, but you've got the same genetic code in all the cells. And obviously what makes a nerve and what makes a cheek cell are very different in terms of switching genes on and off, but the structure of the genes is the same. So we can harvest our genes typically this way. We don't need to go anywhere near the brain. And what we can either do is what's called a genotype first approach. And that's we say, I'm interested in this particular gene. And then we look at that gene and we see differences in behavior. So we've already seen examples of this. So for example, there is a genetic difference in the oxytocin receptor. That is just a single DNA swap. If you will, and you can effectively genotype everybody in the room and say you're type A, you're type G, and then you can give them questionnaire measures, you can look at their mental health and so on. That is a genotype first approach. A phenotype first approach, so let me give you an example of a genotype first approach. So this is another example here, which is looking at a dopamine receptor gene that exists in two different kinds. So you can call these mutations, but sometimes they're so common that basically, they're just different variants. But what you find here is that one particular variant, let me figure it out, the long variant here in green, is associated with greater risk-taking, novelty seeking in both social and non-social situations. So these people are going to enjoy roller coasters more, for example, but also they're more likely to experiment with drugs, for example, with these. So this is a measure of personality. A phenotype first approach, what you do here is that you take a phenotype, and that might be something like empathy, or you take something like autism, and then you search through the entire genome and see what correlates with empathy or what correlates with autism. And of course, the advantage of this is that you're looking across multiple genes. The disadvantage is that your genome is so huge that you need very large samples, and it's hard to get things coming out significant. So here, this is the genome, and it's looking at autism here. So you've just got different color chromes here. These are called Manhattan plots, see if you can figure out why. What you've got on the y-axis is your statistical significance in units of to the power. So 10 to the power of here minus 4 is 0.0001, so to the fourth decimal place. To be considered kind of significant here for multiple comparisons, or people often take this threshold here of 5. And basically what you find is that the more participants you come in, the more these ones here that are approaching significance just get pushed through. So the larger our samples they get, the more genes are being discovered. So we're constantly finding more and more genes for autism the more we look, simply because you're pushing things over the statistical boundary there. So in this study, there's kind of, you can count them on your hands. But again, we don't really get a handle on what these are doing. You can look at how these genes are kind of expressed in the brain, what they code for, these sorts of things. But it might be that this gene is just involved in switching all these genes on. It becomes very hard. And as soon as you've got one story, the story changes because you've discovered a whole other set of 10 genes just by collecting all people. So that's how you kind of can get at linking between exact genes and behavior. We're also very interested in the environment. We know that the environment is important, and it's not just a question of saying, well, it's half genes and half environment. What we're interested in is how do these two things talk to each other, and we know where they're talking to each other because they're talking to each other in the brain. All of our behavior is triggered by our neural circuits. So the substrate by which our environment and our genes talk is in the actual function and structure of the brain. That's for sure. Whether it's hormones, whether it's receptors, whether it's the growth of synapses, we can debate. So let's have a look at some candidate mechanisms by which genetic susceptibility and environmental susceptibility work. So the one you will have heard a lot of is epigenetics. So epigenetics is basically looking at the way that our genes are expressed. So it's not about changing your genetic code. You're given that at conception, but what you can do is that you can make some genes more or less active. And how do you do that? Well, basically your genes are kind of in your double helix, but this is self-organized into chromosomes and wrapped around proteins and kind of structured in various ways. And what you could do is that you could actually add little molecules onto the double helix that causes it to bend and fold in certain ways that makes certain genes hard to read off or easy to read off, in effect. That is the molecular mechanism here. So there are chemicals that are called methyl groups that I think make them less accessible. And there are other ones that make them more accessible that you're just adding on to your genetic code. Now here, it is going to make a difference whether or not you're looking at cells in the brain versus cells in the cheeks. So epigenetic events might be quite tissue specific. They might be, even within the brain, quite cell specific in terms of certain circuits. This means that it's very hard to do these studies in humans, and most of it is coming from animals. The example in animals that is most well worked through is to do with epigenetic effects of the stress response to do with parenting techniques in rodents. Basically, ignore this graph for the moment. That's coming in a minute. But basically, what you've got here is that the parents engage in different amounts of grooming behavior, and grooming is kind of licking and touching and kind of physical contact with the pups. If a pup doesn't get enough kind of licking and grooming, it gets stressed. And this stress response is an epigenetic event where certain stress receptors to do with, in this case, it's cortisol, so a hormone, but receptors in the brain that respond to cortisol, those particular proteins get epigenetic markers in it. That means that those animals are stressed for life. These epigenetic markers are there as a result of having inadequate parenting. You can also foster the pups as well. So if you've got a pup with a neglecting mother and you give the pup to a caring mother, you will show that it is to do with the environment that kind of matters here. It's to do with the mother's parenting rather than the infant's genome. But the infant's genome is the one that bears the burden of this. In humans, this has also been found as well. So this is looking at the brains of people who died early. And in this case, you've got people who'd committed suicide. So the question here is that some people who committed suicide have had very unpleasant childhoods and so on. Other people have committed suicide and had, to the best of our knowledge, normal or caring childhoods, and obviously other things have gone wrong in life. And what you can find here is that these epigenetic changes are not to do with suicide. So suicide is obviously linked to mental health problems. Yeah, I think we have to take that for granted. It is to do with early abuse here in that particular case, that you have less. This is just some kind of glucocorticoid receptor binding or whatever. I would have to look into that. Don't ask me what it is, but you get the gist. So this data is from humans, and we see a similar thing in rats now. So that's epigenetics. Gene-environment correlations are basically that your genes help you to create your environment. So actually the example I've already given of risk-taking is that people with risky genes create risky environments for themselves. They are more likely to choose to go on a roller coaster. They are more likely to, take extra drink or drugs. They just want to push things to the max, okay? And that is the environment that they are creating. You can also see it, so that's this example. You can also see it in terms of parenting style. So the question here, to some extent, your parenting style is going to depend on you as a parent, your genes, but it's also going to depend on your children's genes as well. So some children might have challenging temperaments or more gentle they don't cry much, whatever it is. So your child will also have their genotype. The problem here is that your children are also genetically related to you, so it's very hard to pull apart your genotype from your child's genotype. Obviously, they only share 50% of your genes. The way that you can do this is obviously in the classic kind of designs of adoption, for example, where you have adopted children. The modern twist on adopted children is obviously assisted fertility, in which you've got other people's eggs and sperm, or you've got other people's literally fertilized eggs that you then have put in you literally from like a month after conception. So it's been even in utero in your environment, not just given to you as a 1-year-old. And basically, you can do this kind of modeling here that basically you can show that your child's genotype kind of encourages certain parenting behaviors in them, that a parent will adjust their style according to the child's phenotype, either to be more, I suppose, caring, a bit more punishing, a bit more whatever it is, depending on the child that they're given. And you could pull this apart with these kinds of designs where you're effectively modeling statistically basically your reaction to the child as a result of your genes and a result of the child's gene. Gene and environment interactions are basically the idea that you might have a certain vulnerability, but that vulnerability only matters if you then have an environmental vulnerability. Okay? Or maybe a protective effects would kind of be the same thing. These are the kind of studies that have been well cited, also well discussed and critiqued. There's still heated debate as to whether or not any of these results are valid, but they are certainly interesting, and I'll go through them. So here, this was the Dunedin study, where they had 1,000 consecutive births, and basically they followed they're still following these children through now into, I think, their 40s, maybe their 30s. At the time, this was very groundbreaking and a massive sample. Now, in genetic terms, it's seen as being tiny, and that's one of the problems with it. But the advantage is having this gold standard of longitudinal data that you can ask. You don't have to ask somebody in their 30s, did you take cannabis when you were a teenager? You can actually go back and find out what they were doing as a teenager with that. You're not relying on these retrospective reports. And basically, these are the findings that they had. So this is a monoamine oxidase gene that's involved in the metabolism of serotonin, dopamine, other things that can exist in a more efficient and less efficient form. Basically, if you have one variant of this, I can't remember exactly which one it is, and you have some childhood adversity, then you are far more likely to engage in antisocial behavior than if you don't. So it's not a question of having a bad childhood environment. It's not a question of having bad genes. It's a question of having both in combination, and the risk gets amplified between them. That's what it means to have an interaction statistically. Here, this is a serotonin transporter that exists in short and long forms. And again, if you have negative life events, it's more likely to result in depression, suicidal thoughts. But again, it depends on a combination of that. If you have both, if you tick both of these boxes, your chances are far more elevated than just ticking one or neither of those. And this is, I've forgotten exactly what this one does. I think it's in brain development. It's also related to, I think, breaking down neurotransmitters. And this is looking at smoking cannabis as an adolescent, and that people report more schizophrenia-like symptoms. So that's just this one here. So the gene exists in two forms, depending on whether you've got the amino acid methionine or valine. Is that what val is? And basically, if you have the valine, both, if you've got double kind of valine genes, and you smoked cannabis as an adolescent, your probability of reporting schizophrenic-like symptoms goes very high. And that's at age 26 here. They looked at various other things in the study. So it was about the critical age of smoking at adolescence. If you were smoking cannabis in your 20s, your risk actually didn't go up in this study. It was a sensitive age is what they reported. So again, it's not if you take this at face value, it's not the simple Daily Mail view that smoking cannabis is going to make you schizophrenic, okay? That isn't necessarily the message here. The message is that some people are vulnerable to smoking cannabis at a certain age, if you believe it, and not everybody does. Why don't people believe it? Well, okay, I'll come back to that, but in terms of a mechanism, I think that these are kind of nice stories, whether they're true or not, because instead of saying, oh, it's half genes and half environment, actually we can understand exactly how these two things are working out and how they're playing out at the level of individual circuits, although that story hasn't been fully told. Why are these results controversial? Well, basically, these are candidate gene approaches where people said, I'm interested in this one gene. What you would hope to do is that if you take a group of people, for example, if you look at schizophrenia amongst, say, 20,000, 30,000 people, 100,000 people, you would want to see this gene coming out in that kind of Manhattan plot, and it doesn't. Always with these particular ones here. So when people take the other approach, you don't get it. And people debate why that is. One possibility is that those other ones that are using the kind of sorting through the whole genome are not measuring the environment precisely. So basically, it might be that you're not going to get it out because it doesn't matter on the gene itself. It only matters on the gene in very precise environments, maybe at very precise points in time. And actually, even if you've got 100,000 people, if you're not asking the right question, you're not going to get the right answers, okay? So we shouldn't just kind of believe the study with the largest N. If they haven't actually measured the environment in the right way, it might not give you the right result. So what I think that there's other work to look at it. People have also looked at these candidate genes in other ways. So for example, you can take people with the different monoamine oxidase variants and you can show them angry and fearful faces in the MRI scanner and show that they will have different response patterns. So again, it's very hard to kind of question that isn't a difference, even if we don't get it coming out with this. I think that the other thing that's been said is that a lot of the time when people are talking about environmental influences, they're talking about bad things happening, but actually, for every person with a bad thing happening, there are at least as many people with good things happening to them in life, and often these are not taken into account. So this is a study here looking at different, what they call kind of social sensitivity genes, which includes some of these other ones that we talked about, so the monoamine oxidase, this serotonin transporter genes. Looking at adolescents, and they're just literally counting up how many of these sensitive variants you have. So basically, if you have, in effect, none of these high sensitivity variants, then it doesn't matter whether you have good or bad parenting. You are incredibly resilient in your kind of outcome. And here your outcome is your ability to, I think this is a questionnaire measure, engage in kind of self-control, self-regulation of your emotions. Basically, you can take good parents and bad parents, you are going to get through it with that. What you've then got are those people who have a high genetic load of sensitivity. And what you find here is that those with a high genetic load, as the other studies are reporting, if they've got bad parenting, they respond quite badly in terms of their emotional responsiveness. But actually, there's a whole other side of the equation, is that if you have these risk genes and you have uber good parenting, I'd be curious to know what that is, but anyway, you enable much better than average self-control and self-regulation strategies. So again, are these genes kind of good or bad genes? I don't think you could label them that way. It depends on whether you've got a good or bad environment. And again, most people, for obvious reasons, are only looking at bad environments, looking at adversity, looking at poor outcomes of mental health. I think it's just as interesting to know why some people go through very adverse things and are hugely resilient, have very good mental health as well. And that's probably worth investing in to understand the people at the other end of the distribution. Okay. Any questions about genes and environments? Let's go to the chat. Nobody got anything? Referenced in this lecture. Yes, yeah, I can do that, Alice. I can send you a chapter from my book. You might have to email me, but I'll do my best. Okay. That's really annoying that it freezes. Okay, right. I'm just going to go back to the. Nope, it's definitely frozen. So let's work our way through the kind of the developmental trajectory. So how do infants come to kind of recognize the people around them and kind of engage at this kind of, I suppose, low-level social processing? So an infant doesn't see their mother or any other social being until they're born, but they hear the social world from inside the womb. And I find these a really fascinating set of studies. But basically, you can take a newborn infant and you can play it, in effect, a tape recording, or digital recording, and the measure you're getting is that the behavioral response is that they will, in effect, suck on a dummy in order to hear the recording. Okay, so you can play different kinds of recording and measure the kind of the sucking rate, in effect. And what you find is that they prefer to listen to their own language relative to other language. They prefer to listen to their mother's voice versus other voice, and they even prefer to listen to a familiar story. So here, the mother has read stories to the infant whilst it is in the womb. Okay, and then she reads the story after it's born versus the mother reading a different story that it's not heard before. Isn't that strange? But what you, if you actually look in the brain, then there are parts of the brain that respond to voices more than other auditory stimuli, but these are actually not specialized for voices versus other auditory stimuli until later. So the voice sensitive regions are not specific to voices at that age, even though the infant can kind of discriminate between these sorts of things. In terms of face recognition, basically within an hour of birth, an infant can respond to a face-like stimuli more than a non-face-like stimuli. So how do you kind of have a face-like versus non-face-like stimuli? Here, well, these are, it's kind of almost like a table tennis bat that you kind of hold to the infant here, and then you can measure the infant's orienting to the bat, whether its degree of following, either with its head or its eyes. And in effect, the infant will orient more to a realistic face than to a symmetrical scrambled face than to a blank. Okay. So does this mean that the infant is born with a representation of what a human face looks like? The idea is, well, probably not, but they would have certain preferences that would then naturally gravitate it towards face-like stimuli, including symmetry, including actually top heaviness as well. So these are here looking at top heavy faces. So an infant would prefer this face relative to that one, okay? And again, that's kind of interesting. So obviously we can see that as a face, but actually that is just as face-like. You know, if you live in an upside-down world, there's nothing wrong with that image. And of course, infants don't really have a strong sense of up or down. They're upside down in the womb, at least before being born. And here that's another, but here they prefer this one over that one there. So again, that's a top-heavy configuration that doesn't look anything like I've ever seen before. But they can't discriminate between these two. So they have as much preference for that thing, which I don't like the look of, relative to that one there. That they're not particularly bothered about this. They show an own race preference as young as 3 months. This is done in Israel, where they're looking at immigrants, including from Africa and so on. And they prefer the sex of the primary caregiver. And again, we use this term preference here, but actually knowing that the infant's having an emotional response, to these things, I think is uncertain. But they choose to look at these things. They can discriminate between them in their kind of orienting behaviour. But the term preference, I think, is a little bit loaded, but it is there in the literature. What about actually discriminating between individuals? Well, there is one individual who the infant discriminates very early, and that's the mother. And again, this is probably, this is to do with the special mechanism that it has heard the mother's voice over and over again. Okay, and the father just doesn't have the mother's status because he is not carrying the infant around in the womb. So the infant can learn the mother's face because it already has learnt the mother's voice. So it can make the connection between a familiar stimulus of the voice and a highly familiar stimulus of the voice with the face. And it doesn't, the infant doesn't latch onto other faces in the same way until many months later. So they can't, so habituation is that you show multiple images of the same face and see whether they get bored. They don't kind of show a kind of habituation to the same face. Here, these would be unfamiliar faces like an experimenter's face or something. They don't show that kind of pattern until 3 months. And about the same time, they would show long-term recognition of other faces. So that might be other family members and this sort of thing. Language seems to be important here as well. So there have been studies done looking at infants' abilities to recognize monkey faces, which is a very bizarre study to do. Basically, what you find here is that infants are as good at telling apart monkey faces as they are human faces. So they don't have this kind of human preference, even though they don't see kind of monkeys in their social world. They're only seeing them in their kind of experimental stimuli. But what you find here is that you can, but they lose that ability to kind of differentiate. But you can regain that ability by giving them labels. So in one study, you see monkeys and you see, say, 10 monkeys, and that one's called monkey, that one's called monkey, that one's called monkey. But instead, if you call that monkey Bill, you call that monkey John, you call that monkey Sarah, the infants will go on to be able to recognize and be able to have better discrimination of monkey faces. They don't lose that ability. So it's almost as if humans become different because we individuate, whereas the others are all a category. And this is partly shaped by language, which is in effect a social environment that you're giving the infant to differentiate. In terms of neural substrates, in adults, there is a region that is more sensitive to recognizing known people, called the fusiform face area. If you damage that at one day, then you can become subsequently prosopagnosic. But that doesn't mean that this region is responding only to faces from day one. The evidence it isn't, that it still has to mature over time. And here what you find is that when you show infants' faces, it doesn't just activate this region, it activates a whole other network of region, including ones that in adults we would say are specialized for language and so on. So in adults, the idea is that you've got these kind of specialized regions that respond more to faces than other kinds of things. But in infants, you've got less specialization, is that the faces might kind of activate these early perceptual regions more, but also they're activating other regions that would not normally be activated in in others, in older children, adults. And here the difference in the arrows is basically that the connections between these regions become strengthened developmentally, as well as becoming more specialized responses. This study here showed that actually face-selective regions mature later than object-selective regions. Whether that's because the task in faces is individuating particular individuals and objects, it's not really so. So eye gaze is another mechanism, and this is important for obviously engaging in communication, that I can communicate with my eyes. I can encourage you to look at something by looking at it, these kind of joint attention or triadic interactions. And these become more important kind of early on in infancy, but younger infants can follow gaze to peripheral locations. Meltzoff has done some really interesting studies about what children infants understand about the social significance of this. And what you find is that if your eyes are closed and you turn your head, an infant will still follow to see where you are looking. But then after 10 months it won't. It will understand that if you're turning your head, that in a way that has no real significance. That you're not trying to engage in kind of communication. So whether this is kind of like a proto mind reading, that they're making inferences about the state of the adult's knowledge, that they can see things that you can't, but when their eyes are closed, even when they're orienting to it, you can't. For example, obviously the infant isn't thinking about it in any sophisticated way, but somehow it is behaving as if it has internalized that kind of knowledge. And again, we find that the circuits in adults that are important for this, so the superior temporal sulcus is involved in kind of taking to eye gaze, it tends to be not the dominant system in infants, that they kind of solve these kind of joint attention problems more with, for example, their occipital lobes and their frontal cortex, and then eventually it becomes more specialized in the kind of the adult region. So again, here it's as if the behaviour comes before the neural specialisation is what they're kind of claiming. Okay, how's this going to play? Ooh, yeah. And this is kind of Meltzoff's famous studies of infant imitation. So here, neonates from supposedly the first few hours of birth can imitate certain things. The one that replicates strongly is tongue protrusion. Things like doing an O-shaped mouth and so on replicates as well. It's slightly freaky, but there you go. It doesn't matter if you can't hear it. I'm thinking about it. Okay. Oh, come on. Looks like some good stuff there, but it's not what I'm after. Let's go back to this. Yeah. Okay, so here, obviously, why do people imitate? It's probably to understand their goals rather than motor mimicry. So again, people have debated, that the term imitation isn't really appropriate here. But what's very interesting is that the infant has to match its own motor movements with a visual kind of stimulus. The infant has never seen itself, okay? So to understand that somebody else is producing an action, the sight of somebody else's tongue is related to their own motor actions. It is kind of sophisticated, I would say, despite the fact that people kind of downplay that the infant doesn't understand. It's not true imitation. It's not, not game playing or this sort of thing. And Meltzoff kind of argues that this is kind of like an early like-me hypothesis that the infant kind of has in the world, that there are other people out there like me. And this is the kind of the evidence that he would have, whether it's kind of full-blown imitation, such as the one we've talked about or not. And at some point they can tell whether they're being, whether somebody's imitating them, so where the roles are turned here. And you can study this also with crying behavior, that infants cry more if other infants are crying. And they're also more likely to cry for a tape recording of another baby than a tape recording of themselves, which again, people have kind of debated why that is. The idea is that they can make that sort of discrimination. So Meltzoff and Desity, who are both at the same university, have argued that this kind of imitation is kind of the building blocks of later empathy, in effect. They argue that, that what is functional at birth is not really empathy, but is what they call affective arousal. So certainly you could think of that in terms of the contagious crying studies, for example, that the infant can pick up on, you know, good and bad vibes, if you will, in the environment, and can kind of respond to those. And then what you would have kind of during infancy going to childhood is a kind of emotion understanding, if you will. So what the significance of emotions are, again, linguistic labeling of emotions, these sorts of things. And then what you would have in later childhood and adolescence is emotion regulation. So again, not involving the emotional brain, but probably involving your frontal cortex to actually reevaluate your emotions in order to switch them off, to reappraise them, to rethink things, to in effect feed all the way back down, to stop being affected by emotions, to respond coldly. But sometimes that's a good thing. You know, if you're in a disaster situation, you don't want to go to pieces. You're going to have to keep it together. Yeah. What about mirror neurons and these kind of other mechanisms of imitation? So Cook et al. Have argued that the mirror neurons are in effect learnt. They're not innate, and they're learnt via simple associative learning. So in a way, they're kind of, they're not kind of completely against the kind of the Meltzoff ideas, but they say that is how mirror neurons are formed. That they learn that other people are like them. They would eventually see themselves in a mirror, and they would be able to then understand their own actions are kind of mirrored in others. So here the idea is that they have separate visual systems and separate motor systems, and mirror neurons simply do not exist. And then through experience, what happens is that as you grasp for an object, you see the visual consequences of your own actions. As you engage in these kind of social interactions, you've got mirroring and imitation there. You've got mirrors themselves. You've got social activities that you kind of build up your mirror system repertoire, that in effect you end up that when you see somebody grasping, you activate your own motor system. So their claim in effect is that mirror neurons are not social. They don't exist for social reasons. They are a byproduct of more generic learning mechanisms. I struggle a little bit with this viewpoint, because obviously, even if they are learned via generic learning mechanisms, that doesn't mean that they don't have a social function themselves. Others have certainly argued that mirror neurons are there from birth. So this is a very cute photograph, or maybe weird, depending on where you look at it. But again, macaque monkeys here with a little EEG cap on will engage in imitation of the tongue. They don't engage in more complex imitation, you know, even as adults. But they show this kind of mu suppression. So remember mu suppression, we saw this with the autistic kind of children when they see hand actions, that they don't get this mu suppression, but when they do hand actions themselves, they do. But you can show that this mu system is in place in macaque monkeys from birth, okay? With that, and suppression just means that, so here this is a motor action, and this is observing a motor action. I think these are probably just different electrode positions. What do we make of that? Well, Cook et al. Do kind of talk about this sort of evidence and say, for one thing, the link between mu suppression and mirror neurons isn't proved. Okay, that's what they would say. And they would say that sticking your tongue out is not true imitation. That's what they would also say. So again, I don't quite know how to resolve that debate. To me, both views have their own merits. So certainly some simple actions of imitation are present from birth. At 9 months, so this is in human infants, that you can show that mu rhythms are able to discriminate goal-based actions from meaningless movements. The question is, why not before that? Do you have to kind of learn it? I don't know. And again, we've got these kinds of studies in infants that they imitate goals rather than actions. So here what you see is that the adult switches on a light, and in one case they switch on in both cases they switch on the light with their head, but in one case their hands are free, and in the other case their hands aren't free. Okay, and here it's as if the infant only is inferring that when the hands are restrained, that the adult would have pressed it with their hands if they weren't, that they kind of use their hands even when the person has used their heads, because they're inferring the goal. So again, it suggests a kind of a sophistication at around 18 months in terms of not just motor mimicry, that between kind of sticking your tongue out to these more complex goal-directed actions, that this all happens in infancy. But this is also quite problematic because the classic studies say that theory of mind happens around the age of 4. Okay, so now we've got a puzzle as to these kind of sophisticated goal-like behaviors that some people would say require some kind of theory of mind, and the passing of theory of mind tests at 4. So what is it that's happening? Is this theory of mind? And you know, we just can't ask an 18-year-old to put it in words, or is it something different? Okay. So let's have a break for 10 minutes, and then we'll come to answer that question, and we'll move on to adolescence. I will put the attendance pin in there, and we'll start again at about 5 past. Okay, let's make a start. So we'll just finish off with theory of mind and then adolescence. So here, this is the puzzle that I kind of left you with, is that at 18 months, infants are able to understand intentions on these kind of imitative tasks, and they can distinguish pretend from real. So why does it take until 4 years to pass the classic theory of mind test? So these are the classic theory of mind tests. The kind of hiding an object in one location, the person sees it, then the other person leaves the room, the object gets moved, then they come back. Where is she going to look for the object? Where does she think it is? And so on. The children don't pass this until the age of 4 years. So what's happening between 18 and the age of 4 or 5, 18 months and the age of 4 or 5? So one possibility is that other skills are kind of needed to catch up. So actually an 18-month-old has quite good language. They have good understanding of language, even if their ability to produce it is more limited. Certainly executive control, so your kind of frontal lobes holding in mind multiple options, suppressing reality, all these things, is going to develop. But actually, the textbooks say that that's a kind of more adolescent kind of development rather than an early childhood, but it's something that's probably going to mature all the way. It's possible that there is just something special about false beliefs that's quite different from somebody turning their head with their eyes closed, or somebody pushing a button with their head and not their hands, and all this sort of thing. That's also possible. There is also it's also possible that there are different kind of forms of theory of mind. So people talk about an implicit versus an explicit form of theory of mind that kind of is there in 18 months, and that actually it kind of changes character during development. Or the alternative is a two systems account, that you can have an implicit theory of mind from 18 months and your explicit one develops somewhat later off the back of it. This is a study that really threw the kind of the cat amongst the pigeons in terms of false beliefs and a theory of mind. Well, there are a couple of studies. This one was actually done at Sussex before, and showing that 3-year-olds in the kind of the classic Simon Baron-Cohen kind of studies here, that they would look at the correct answer, even though they stated the incorrect answer with the physical reality of this. Okay. Then this study did something similar in 15 months where there were no language at all. So here there was a narrative as well as a play, if you will. Whereas here you've got somebody who is looking at 2 boxes, and I don't know why, but there is a watermelon that goes into one box and gets moved as if by magic from one box into another. So here the person can see the watermelon move, and then you've got various scenarios. So here in which the watermelon moves back, but a screen comes up and the adult can't see the watermelon change back in the boxes. Then what you have is that the adult reaches into one of the boxes to get the watermelon. And what would be surprising, or what an adult would expect, is that they will reach into the box that they believe contains the watermelon rather than reach in the box that physically contains the watermelon. Okay? What you find is that infants are very surprised when the adult reaches according to the true reality against their beliefs. Okay, so it's as if the infant is surprised when the adult gets it right, and it suggests that the infant understands that the adult knows that. Well, is surprised that it hasn't moved. I don't know if I explained that well, but that's the key essence: is that that the adult's action matches their false belief. The infants look less, okay, when it's a false belief. So here, this is suggesting that 15-year-olds kind of have the abilities of false belief tests in this scenario. So what's going on? So one possibility is that there is some what I call domain general developmental catching up. So with regards to language, with regards to kind of holding in mind multiple response options. Again, it could be the idea that there is an implicit understanding of mental states, but not an explicit one, and that this is what's doing it. What we don't quite have is a kind of a developmental trajectory of how the different brain regions, which we know in adults are involved, come online. What we find is that EEG studies show that the temporoparietal junction and the medial prefrontal cortex, if you apply these at in children at age 4, so those who can pass it versus those who can't, that you do have differences in these kind of adult-like regions. So here the children are developmentally matched, but some have just reached age match, but some have reached a developmental milestone that others haven't. And you could show that there are differences there in these explicit measures. So it seems that maybe that these brain regions are tracking kind of an explicit understanding of that. We don't really know what brain regions are involved in this kind of these looking-based or kind of more sensory motor-based theory of mind or imitation or whatever. I don't think we have a clear answer to that. Again, here this is just showing that there is a link between verbal mental age and passing, in this case, the Sally Ann task, and the other one's the smartest task, where you've got sweets inside a tube. It's a pencil inside a tube, but the child says, oh, it's sweets. And then you say, well, what's the other child going to think? And some child will say they're going to think it's sweets, and the others will say they're going to think it's a pen. So this is the ability to do that here. So verbal mental age does matter. But again, you can have differences. So even when you equate for verbal age, so if you've got a verbal age of 60 months, so what's that, 5 years, for example? So at the age of 5, most typically developing children are passing both false beliefs, but autistic children aren't. So it can't be just verbal age that matters here. So this is an alternative account by Butterfield and Apley. I think they probably need to think of alternative labels for this, but they call them System 1 and System 2 in effect. And they argue that there are two different mechanisms for theory of mind that have different neural substrates and different maturational profiles. So system one is this kind of infant-like system that enables belief-like behavior based on gaze, action, etc. And this permits a kind of intuitive or implicit theory of mind, and this develops early. So it's what we see in this kind of weird watermelon study where the watermelon moves between the boxes. And then there is another system that is based on kind of more explicit beliefs, develops late and is cognitively slower, rather than intuitive. So they argue that this is the early developing, this is the late developing. In terms of autism, they would say that both systems are often impaired in autism, but actually we can see that that some people with autism with good verbal intelligence and adults with autism often pass these theory of mind studies. But actually, adults with autism don't show the surprise pattern with their eyes. They don't show the looking behavior in false beliefs, even though an 18-month-old typically developing infant will. So according to these people, they would argue that it is this early developing system that is impaired in autism. And to some extent, you can get around it by developing this other system, but obviously this system is helpful for developing the other one. So again, it depends what kind of evidence you look at, whether you look at kind of behavior or whether you look at reasoning and kind of labeling beliefs and knowledge and so on. So to summarize this, early competences and preferences, e.g. for face-like stimuli, drive subsequent specialization. This specialization is not present early on, and to some extent it is kind of experience and behavior that helps almost certainly helps these systems to mature in the brain. What are the seeds of things like empathy and theory of mind? Well, certainly imitation, emotion contagion, so this is the kind of crying and so on. Sharing crying is almost certainly going to be important. And we still have this puzzle about how theory of mind develops. And whether or not the developmental evidence suggests that there are different mechanisms, or whether it's the same mechanism that just goes kind of early and late stages of maturation. Okay, so let's move on to adolescence. So the onset of adolescence is quite easy to define biologically because these are in effect puberty. So obviously easier to define in women because it's the onset of your first period. In effect, in men, I'm not quite sure how it's defined, but again, hormonal changes and so on. Here, what's going on in adolescence socially is probably less influence of the family and more influence of peers. A kind of an interest and a concern for your identity, so how you see yourself and your reputation, how others see you and how you want others to see you, that these become very important considerations to adolescence in a way that isn't like that before. And we can think of this as more a kind of a motivational stance rather than capacity. It's not that that younger children can't think about their reputation. It's just that it doesn't bother them in the same way necessarily. And for whatever reason, adolescence, it does bother them, whether it's sexual maturity, other reasons we can kind of unpick. What you do find changing, and these are questionnaire measures, are impulsivity is something that characterizes children more than adolescents. So this is impulsivity here. So an impulsivity item in this questionnaire is, I think that planning takes the fun out of things. Okay, so this is something that is kind of just kind of appears linearly over time. This is sensation seeking. So I enjoy new and exciting experiences, even if they're a little frightening or unusual. So this shows a kind of a peak in adolescence that children are, younger children, what's this? Well, 12, so just pre-adolescent here, tend to respond less favorably to that. And then as you go into mid-20s again, it's less exciting to be frightened or do unusual things. So this seems to be a particular behavioral characteristic of this age group. Here, this is a classic kind of risk-taking study, and that's been looked at behaviorally and in terms of the brain. Hopefully it will work. How's the volume?
I'm about to make a life or death decision myself. Fortunately,
Oh, I didn't mean to do that.
I'm at Temple University in Philadelphia, playing the stoplight game.
I don't think so. It just saved my life.
We've all had the experience of trying to get someplace in a hurry, approaching an intersection, hitting a yellow light, and making a last-minute decision about whether we want to go through or not.
Right.
And that's what this does.
Well, I would have made that for sure. I'm gonna go through this. I made it again. Oh, no, okay. Oh, so I get this broken windshield. That's probably my head that made this.
A more typical participant in the stoplight game is a teenager playing like Alex here while lying in an MRI machine.
He made it through the first one without getting killed. He seems kind of lucky. I would have stopped.
Alex is deciding whether to put speed ahead of safety while he's alone in the scanner. But that, as far as Alex believes, is about to change.
Hey Alex, how you doing? I have your friends here, and they're gonna be watching you play the next round of tasks. So they're gonna talk to you a little bit, okay? Alright, you guys have some fun.
Hey Alex, you still alive in there? Oh yeah. Nice.
Hey Alex, it's Paul. How's it going? I'm good, man. Good luck, and they're gonna be watching you.
Actually, they won't. But Alex doesn't know that. As far as he's concerned, his friends are watching his every choice.
Crash. Bang. Is it my imagination, or is he having a few more crashes this time?
No, he is having more crashes, and that's what we find when adolescents play this game. They crash more often when they know their friends are watching them than they do when they're by themselves.
What happens with adults when they're with friends or alone? Is there that much difference with adults?
There's no difference at all.
Really?
In how they behave or in their patterns of brain activity. So when adults are alone and adolescents are alone, they look very similar. It's when you put adolescents with their friends that their behavior and their brain pattern changes.
The brain pattern change is in the regions that respond to reward, principally in an area called the ventral striatum. When young people are alone playing the game, the ventral striatum remains quiet. But with friends around, it lights up.
During adolescence, peers are so important that their presence activates the brain's reward circuitry, and that makes adolescents, under that condition, pay more attention to the potential rewards of a risky choice and less attention to the potential downside.
Oh, bravo, you did a great job. Your friends are proud of you. Just don't get in the car with her.
Okay, that's probably enough. Okay, so again, this is obviously a fairly contrived situation. So, but I'm sure that the insurance statistics and so on kind of back up this. I think we could all believe it. Yeah. Okay. Oh, I see. Okay, it needs me to get to the end of the video before it will allow me to move on.
And this play, which I think the document now.
All right, okay, right, yeah, let's move on. Let's see if I can do that again. Oh, there we go. Yep. Okay, let's have, right, so this is just the paradigm here, okay? So it's very easy. You saw what he was doing. He was just pressing the space bar when he gets to a yellow light to stop or not. If you stop, then basically you get a 3-second delay. So the motivation here is to get around the circuit as fast as possible, okay? If you go and you make it so the car doesn't hit you, then you get no delay, okay? But if you do, then you get a 6-second delay. So here it's kind of weighted, I suppose. I don't know whether these are 50-50 equal, but on average they're equally there. But these are risky, whereas this is a known, it's not risky, it's a known delay in effect. And what you've got here is a number of risky decisions. So that would be go through an amber light. And you can see in the blue bars that adolescents, young adults and adults are exactly the same when they are alone in the car. But when a peer is present, it makes a big difference on the adolescents. And it's said here, it's kind of it's fictitious. You know, the peers aren't necessarily present. I mean, they're in an MRI scanner, for God's sake. You know, so it's like they're not driving a car. And this is the number of crashes. I mean, that's just, you know, going to happen in the same way. What's interesting here is that you can play the game, you know, in the MRI scanner and show what's going on. What you find here is that your prefrontal cortex, your lateral prefrontal cortex here, matures over time, but actually is not the thing that is driving the interaction. So adults activate the prefrontal cortex more in the driving game than young adults, and young adults more than adolescents. Okay, so this is just a kind of a general kind of control over the situation. What you then have is the interaction appearing in the reward centers of the ventral striatum, the orbitofrontal cortex, that is specific to the adolescent group in the presence of their peers. Okay. We can debate what that means, whether or not the peers themselves are rewarding, whether the peers are kind of, you know, increasing the rewards of the game, and the two are kind of becoming conflated, and that you become almost blind to the downside of this. So it's kind of a high weighting of social rewards. Who knows why? Here you've got another one where you've got, let me remember what this is. So this is where how influenced you are by other people's opinions. So you're given scenarios like crossing a street on a red light, how risky that is or not. And you're told how you give your rating, then you're told how other people rated it, but you're told how adults have rated it or how teenagers rate it, and then you form your own opinion again about that level of risk. Okay, and in effect, this is kind of a degree of modulation from the baseline according to whether or not you've been influenced by the group speak, if you will, of the risk. And what you find is that everybody's influenced by group speak, but people here, the younger adolescents are more influenced by teenagers, whereas older adolescents and adults are more influenced by other adults, in effect. So which group you kind of pay attention to is somewhat age dependent. You might wonder how much of this is kind of social or cultural, but actually teenage rats also like to get drunk with their friends and their brothers, okay? So they don't have a kind of a Friday night kind of West Street culture or whatever going on here. So what you've got here is that you've got an adolescent rat, basically, who they have a peer come in, and the peer's breath either smells of alcohol, it smells of coffee, or it smells of nothing. Okay, and then the rat can choose whether or not it wants to drink alcohol or whether it wants to drink coffee. Is that right? Yes, exactly. That's right. So the rat has a choice of alcohol or coffee, and the demonstrator, who's one of its brother, is either drunk or been on coffee or been on water. And an adolescent rat will choose to drink alcohol when one of its siblings, when it can smell alcohol on the breath of one of its peers. And again, this is somewhat age dependent in this kind of mechanism. So again, there is a link between social disinhibition, kind of following the crowd and so on, whatever you want to call it, rewards of alcohol, that is somewhat independent of culture. You see it in other species when you introduce alcohol to them. So what's going on in the brain? So myelation increases steadily from birth, but it shows a different kind of profile in different regions. And the prefrontal cortex is the one that shows the last region to kind of achieve adult levels of myelation. People's brains are kind of adult about the age of 23, something like this. which is kind of interesting if you think that, the legal age is 16 or 18 and people are capable of having children at the age of 13. So why is there like a decade between your brain maturing and the age at which you're capable of having children? And again, that might be a good reason that you're willing to take those risks. And that's a good thing for the survival of the species. Whether it's good to have teenage pregnancies or not, we can debate. But there is a kind of a lag between sexual maturity and brain maturity, that's for sure. And grey matter shows a rise and fall pattern again. So these are just different studies that are looking at grey matter volume and white matter volume. So your grey matter goes down. That's partly because as your grey matter becomes myelinated, it just changes its colour in the MRI scanner. It kind of almost flips its status for some of this. So the idea is that the myelination from the frontal lobes enables you to engage in cognitive control, in this case when you're driving a car, but also enables you to suppress risk, to regulate your emotions, all those kinds of late developing social behaviours. One theory around this is that the mismatched development, if you will, of different brain regions is not a coincidence. It's kind of a kind of, well, I hate to say the word design because there isn't a designer, but it is something there to promote a particular set of behaviours in this age group. So the idea is that you've got almost a seesaw kind of between between risk taking, which is kind of more approach reward sensitivity, versus the consequences of risk, punishment, getting things wrong, and so on. And that in effect, in adolescence, it is balanced more towards rewards and kind of approach behaviors rather than avoidance. And that this is the kind of adult pattern where the seesaw is more balanced, that you have a kind of, well, I don't know if it's a more healthy approach to risk or not, but you have a different approach to risk in adults, particularly social risks. With your parts of your frontal cortex, so I don't know why it's labeled medial ventral here, that would be your kind of more reward-centered frontal cortex, and then you would have your lateral frontal cortex, which is less reward-centered, kind of on top of that as well, helping to restore this balance. So you could show that there are these differences within these reward and perhaps punishment-sensitive regions of the brain. That the amygdala kind of increases in childhood, whereas the nucleus accumbens kind of shows a kind of reverse profile here. How do adolescents kind of perform in things like mentalizing tests? So behaviorally, we would say that kind of theory of mind and so on is pretty much mature at the age of 4. So what's going on in adolescence? Well, obviously more complex mentalizing tests. So John thinks that, James thinks that, Sunny thinks that. These are late developing, that probably require kind of more reasoning, more executive functions, and so on. What you find is that adolescents involve more of the kind of more of their frontal lobes and less of, for example, the temporoparietal junction that others have. And again, this is interesting because people like Rebecca Sachs argue that the temporoparietal junction is kind of a uniquely human kind of modular theory of mind region. And if it is a modular theory of mind region, it comes on gradually through development. It's certainly not something that appears specialized early on. And this is kind of a complex theory of mind task, where here you've got this person there who's giving you instructions, and the instructions are move the small ball left. Now, the key thing about this is that there are in fact, there are 3 balls here, but this person cannot see that ball, okay? So the correct answer is to move this ball left. And that's, in effect, a kind of a foil. So which is the small ball here depends on whether or not you take your perspective, in which case it's that, or you take this person's perspective. And actually, this is something that people are error prone on. So adults are still kind of 50% kind of error prone on these sorts of tasks. This is a control condition here. Adolescents are, 70% kind of error prone on these sorts of tasks. I'm not quite sure why it's so high. I have to say it must be more difficult. Maybe it's done under kind of time pressure or something like this. But the idea is that these kinds of skills are still maturing in adolescence. They're not all there at the age of 4, even if a basic capacity that this person sees things differently from you is present from 18 months, if not 10 months. That the idea that we have different knowledge based on our different viewpoints is very early developing, but that doesn't mean that we can accomplish this in the real world in sophisticated contexts until later in life. Any questions about adolescence? It's kind of quite interesting, isn't it? I really like that driving study. Okay, so adolescence is associated with increased risk-taking, but this is quite context-specific. And particularly, there's good evidence of a social influence of peers. Changes in the prefrontal cortex is likely to be important both structurally and functionally. It's kind of a form of emotional regulation, but there might also be differences in terms of reward sensitivity as well, and that the two are kind of almost in opposition to each other, that rewards are very salient and your ability to regulate those rewards are not fully developed. Okay, so good luck for the rest of your masters. I've really enjoyed teaching you. I'm still around to kind of answer emails. I won't be around for about 10 days over Christmas, so don't expect an instant reply for that. I'm working my way through your reports as well. Okay.